Welcome to Portfolio Stories, Science and Success, a podcast where we explore inspiring journeys, groundbreaking research, and the people shaping the future of science.
Hello everyone, welcome to Portfolio Stories, Science and Success. My name is Raluca Contu and I'm a postdoctoral researcher at Łukasiewicz Port. I'll be your host for this special episode recording during the 17th International Congress of the Polish Neuroscience Society here in Wrocław. Today, we are joined by Dr. Mathias Schmidt, head of the Stress Resilience Research Group at the Max Planck Institute of Psychiatry in Munich. His research investigates the molecular and behavioral mechanisms of stress vulnerability with a focus on neuropsychiatric and metabolic consequences. By combining advanced mouse models with automated analysis of complex behavior, his work bridges basic neuroscience and translational insights that could contribute to the development of future treatments for stress-related disorders. Mathias is also a valued partner of Łukasiewicz Port and played a pivotal role in the implementation of the same NeuroID project, which led to the foundation for further collaborations such as the AutoHealth project or the Astromics project. Mathias, thank you so much for agreeing to be here with us today.
It's my pleasure. Thank you for inviting me.
So let's start our conversation. I would first like to ask you to tell us a little bit about your research path and how it shaped the current focus of your lab.
When do you want me to start? Very early or?
Well, maybe around PhD. How about that?
So at this time, when I was doing my master's thesis, which happened to be in the United States at the University of Delaware with Gig Levine, at this time, I really fell in love with stress research. So he was an eminent scientist studying early life stress and stress physiology and stress development. So at this time, I told him, look, this is something that I would like to pursue. And I really liked working in his lab and then everything. And then I talked to different people and he advised me to go to different labs. And I ended up doing my PhD in Leiden at the University Medical Center in Leiden under the guidance of Ron de Kloet and Melly Oitzel at this time. And then after having done my PhD, also then continuing to study the development of the stress system in mice and the first gene by environment interactions, basically, I started my postdoc in Munich at the Max Planck Institute of Psychiatry, where then after a while I was offered the chance to head my own research group and then continued into studying stress further with a focus on stress resilience and susceptibility.
Yeah, I can see that you had a quite international experience.
Yes, so I started going to the US because I wanted to get a different opinion or a different experience during my studies, and then really liked being in a different place and being in a different environment and learning also from international mentors. So that really always helped to find my path and be able to pursue a passion or something that really started to interest me. Yep.
Thank you very much. So I see that your projects address stress exposure at different time points in life. And if I'm not wrong, your recent work highlighted that early life stress affects males and females differently.
Absolutely.
So can you elaborate about your findings?
Yes, maybe also let me start a bit historically, right? So it is long known that stress-related disorders are occurring with different frequency and are more predominant either in men or in women, depending on what kind of disorder you're looking at. But in preclinical research and translational research, when I started with my master's, also with the PhD, the female animals that could have been part of the experiments were usually excluded. This was the way of the field, and everyone was basically telling me and themselves as researchers that yes, when we study males, it's just much easier. We're not having to deal with the cycle of the females, and it would be much more difficult. But basically, we can extrapolate everything that we see in males to females, and then we basically also know how things are working. And I think deep down, a lot of people knew for a long time that this cannot be true, right? But it's only recently that this has been embraced and then studied more carefully. And I must say that even though the sex differences have been expected, and we know from epidemiological studies that there are going to be sex differences, the magnitude of the differences that we find, not only on the phenotypic level, but also on the underlying mechanistic level, structural or connectivity, or no matter where you look, you see so many differences, even if they may lead to the same phenotype, which then really, like, basically what follows from that is that the need to treat stress-related disorders and also pharmacologically address specific symptoms you want to change, like you would like to do in a psychiatric disorder, need to be sex-specific, need to be different in women and men. So I think this is super important, and I'm happy that our data can contribute in this direction for more insights that we're getting.
So do you think that these sex-related differences are coming out because there is a different pathway that is involved in male and women in men and women from when we perceive stress to when the outcome is there? Or do you think that the pathway itself is similar, but then the outcome is different?
I believe both can be true. So there's going to be instances, and there's been some beautiful reviews on that, where the outcome is apparently the same, so a similar type of behavioral change, for example, but the mechanism leading to this might be very, very different. There's other instances where a similar mechanism in the cells or circuits in the brain, for example, may then drive very different behavioral adaptations, because the wiring of the brain, because of hormonal influences, because of history of the individuals, because of social constructs, all of these things may play a role. And that's why I think if it was only the symptomatology which was different between women and men, or males and females, and the underlying mechanisms would be identical, then it wouldn't be such a major issue in the study of stress-related disorders, because then you would be able to, with the same drugs, for example, treat the same mechanism and get maybe a different symptomatologic outcome. But it's not that simple. Often it's very different mechanisms, different brain regions, different circuits, different targets, which would need to be addressed in women versus men. And maybe I just want to add to this now, we're thinking of sex as a binary concept often, because it makes it easier to study and also to address. But the reality, again, as we know in humans for sure, but probably also in animals, is not as straightforward and black and white, and there is more like a continuum, and especially in humans, but then also in our animal models, we should also be studying the situation, not just in a black and white male-female situation, but more as a continuum, to also include issues that we know we have with gender minorities, for example, in humans.
Yeah, so I'm glad that you mentioned the humans and the animal models, because I believe that is also one of the details that we need to tackle when studying stress. So how are you linking the findings from mouse models to the human cohorts and to the clinical data?
Yeah, that's a really
to bridge the translational research gaps.
Yeah, that's a really important point, because in the past, the scientific community as a whole was not super successful in bridging this gap, and has been termed the valley of death of translation, right, from the preclinical side to the clinical reality. We, as again, preclinical researchers, have cured depression and other stress-related disorders hundreds of times in our models. Very little to none of that has actually gained any traction in the clinical reality and treatment reality. So there's different reasons for that. One that we're working on quite heavily is an improved phenotyping of our animals. They are excellent model systems. Rodents specifically are very good model systems, but the simplification of readouts and the oversimplification of their interpretation, I think, is a major issue. And a lot of times in the past, we and others, so our lab included, have taken some interpretational shortcuts, labeling a given phenotype as pathological or disease-related, while there was no real good evidence for that. And with the advances of machine learning and AI and modern technology, we are now much more able to integrate really holistic behavioral phenotype of animals in their natural environments or semi-natural environments over longer periods of time, and then generate a lot richer understanding of their behavioral repertoire, similar to what we would have in a human condition. So the alignment of data that we can obtain from the animals and humans, I think, is super important. And this is happening on the behavioral level, but on many of the other organizational levels as well. So both, on the one hand, in the clinical research and clinical studies, methodology, of course, is improving. So we get a lot better resolution of brain activity, for example, in patients versus controls, or genetics, epigenetics, giving us a lot of insights. But of course, still we're limited in the humans with access to tissue, access to experimental treatments, or we can't try to make anyone worse, right? So with that in mind, the preclinical research is really, really helpful in elucidating mechanistic underpinnings, but always trying to match our readouts and match the possibilities that we have with phenotypic analysis or mechanistic analysis to samples that we can also obtain from humans. So one example is that we can look at gene expression changes and then link those gene expression changes to expression changes that have been obtained in postmortem tissue, for example, from suicidal patients with or without a history of depression, for example, or PTSD, or what else.
Yeah, I completely agree that with the recent advancement and automated behavior analysis of animals, and also in the multiomics, we can now have a bit more relationship between the animal models and the actual patients. So based on your findings, how do you envision the novel pharmaceutical or non-pharmaceutical interventions to boost stress resilience in humans?
That's a good question. Let's start with the non-pharmaceutical interventions. So we are learning a lot every week, every day, every month, and we're working on and understanding stress system resilience, susceptibility. We are learning a lot over the time, and some of the things that we are understanding and learning will give us tools to potentially intervene, which we can then test, or can be tested in humans. So one example where we are working on is autophagy, which is basically the machinery of waste disposal and degradation of old organelles and proteins in cells. It's very, very important, and we have getting more and more better understanding how this machinery and how this process of autophagy is integrated and essential for also stress functioning and consequences of stress, all the way leading to psychopathology. And the good thing about autophagy processes in general is that we already have some good evidence to how to boost, for example, autophagy. So we know that exercise can do this. We know that certain types of diet or diet interventions can do this. So we have now tools in hand that we can test whether these can actually promote resilience or reduce susceptibility to stress-related disorders. And from a pharmacological point of view, again, we need good targets to study this. And with our research, we are finding additional or novel targets which can then be used as a treatment target for intervention. What I think is super important is the specificity of treatment. So one target which in one cell type may do one thing, may do something else in another. And there again, we are reliant on technological advances which are in the process of being developed, I would say, which then enable a very specific targeting of a given drug or a target delivery of a given drug to only a region of interest or cell type of interest at a given time. And I think with these fine-tuned advances, we also have the possibility for precision medicine, which we're gonna need for treating these complex disorders.
So you think that moving forward, the medicine in the context of stress-related and neuropsychiatric disorders will have to be tailored to a particular region of the brain or to particular cell types that are more involved in the phenotypes that we are seeing.
Yes, I think this would be ideal, because otherwise we will always get side effects. Most of the drugs that we are currently taking are acting everywhere in our bodies, and if you have a widespread target, then you get widespread effects. The net effect may still be positive, but you get side effects that are unwanted, and biology, as we always learn, is not black and white. And every risk factor can also maybe contribute to resilience in another context, for example, or in another cell type, right? So targeting specifically the regions and cell types of interest, I think, is going to be important.
Thank you. So I remember during the COVID pandemics that there was a dramatic increase in anxiety and depressive disorders. So given these recent global challenges, how has your work on stress resilience during prolonged stress have changed? Have you received more renowned significance, or how did the pandemic change the vision of your work?
Well, I think one thing that happened during the pandemic is that it, like, the society and also on every personal level, people were forced to be challenged with this additional stressor, and this showed on a global level basically what we already knew before, but now in a very convincing real life experiment, if you want, that the consequences of stress can be quite detrimental, and we saw this in the increase of psychiatric disorders, stress-related disorders specifically. But unfortunately, I mean, this pandemic is only one of the many stressors we're currently facing, which are novel, unprecedented types of stressors also. And it's not necessarily only stress generally is that thing that happens all the time, right? And we are very able to deal with this, and our bodies are very well equipped to deal with this. But it's the controllability and predictability of stressors, which I think is crucial. And there are, unfortunately, in many parts of society in the world, a lot of high and unpredictable stressors, often man-made, which is unfortunate. And these will have dramatic consequences on human health, not only for the current generation, but also for generations to come, right? So with this, I think, this put the spotlight of stress and stress-related disorders even more in the foreground than it was maybe 5 or 10 years ago before the pandemic. And yes, it just urges us on even more to find the mechanisms governing this and to be able to counteract.
So I think that one of the key words and the important points that you just mentioned, and I think it was also mentioned today at the Congress, is this controllability of the stress response. And I think our audience here is very interested in that. So do you think that this controllability is biological, or do you think that we have the power to control it? So if I don't know, see, I have the stressor right here, the threat. Do you think, what do you think it's stronger, my willingness to deal with it, or my, I don't know, genetic background or biological background?
Well, again, that's a complex question, and the answer is maybe not so straightforward. But first of all, I would like to say that the types of stressors we're exposed to can vary on level on controllability, just very objectively. So thirst is a stressor, and it's very controllable. And if I'm in an environment which offers me something to drink, right? So if I'm thirsty, I can, not suffer from that stress situation, physiological stress situation, because I can remedy it by getting a glass of water, right? And then there's other stressors which maybe inherently are uncontrollable. The pandemic, especially in the first couple of months, was a good example for that, because there was an external threat to individuals and families and society, which was apparently at that time very uncontrollable, right? Because you had no means to counteract this. Then with the introduction or making people aware that masks can help, that vaccinations can help, then there were tools in place where you could actually manage the stress situation. And at that time, it was also seen that the level of stress-related disorders was going down again following introduction of this controllability. Now, there's, of course, also a very personal aspect to this, right? So what I objectively may call a controllable stress situation for you might be highly uncontrollable. So you could say, oh, I'm very stressed at work. My boss is super mean to me and I cannot get out of this because then this would be a very chronic stress situation for you. Could be a relationship, all kinds of levels, right? While from an outside point of view, one could then say, well, maybe it's not as uncontrollable, right? It may be out of your comfort zone or require more drastic steps that you would like to take, but there is actually ways to get around this. And I think this is something, some type of training, self-training, everyone can do to question how uncontrollable specific stress situations actually are and what tools one might have to actually control this. So some situations you may really, like if you're suffering from a chronic illness, for example, you may be really out of control. But then sometimes also it helps to accept this level of uncontrollability, and if you can't change it, don't fuss about it, basically. So there is a certain psychological training that at least we as humans can do to avoid the negative consequences of uncontrollable stress situation.
Yeah, so it sounds like we also have a word to say in our life. It's not only our genetic background that's...
No, the genetic background, also our history, which often we didn't choose, our parents, grandparents, our time in the womb of the mother and the first couple of months or years of life, none of us had any real control over. So we're coming with this baggage, if you want, of resilience or susceptibility from the genetic side, but also from our life experience point of view. But still, at the time when we are like in the here and now, as young adults or adults, then we can certainly still manipulate or change the course of action and have additional means to tweak resilience or susceptibility. Yeah.
Yes, thank you. Thank you very much for the information. I would like to address a different side of research now. So I know that you have a wide research, a wide network of research collaborators, and across different countries, across different institutes, also different projects. And we actually have a work together on the Ortho Health project.
Mm-hmm.
So how are collaborations shaping your research on stress resilience? How important are these collaborations?
Yeah, they are. I can't stress this more. They're very important because you can. And we are basically at the forefront of scientific discovery. We are trying to understand stuff that we don't understand yet, and doing experiments to understand processes and in our case, stress resilience. We could do it alone. I say we, I could personally do it alone. I would be very slow and ineffective, and I couldn't be checking my results with others. Now, within a lab, there's a lot of people very important, PhD students, postdocs, technicians working together. This is already a benefit because then as a team, you're much more efficient, you're getting more reliable and better results and more insights, but still you're in a bubble in terms of what you can and cannot do, but also your ideas you're having on your scientific hypotheses, for example. So to then engage in collaboration with other scientists at other locations, which have other training, other background, other technological possibilities, for example, is very, very important because it just broadens our view, our possibilities on studying resilience in our case, and is just very a lot of fun also to work as a team and to gain this additional knowledge as a team is I think is always preferable to doing it in a more isolated fashion. Yeah.
So what do you think are the secrets of a successful collaboration?
The secrets of a successful collaboration are like the secrets of a successful marriage.
You will elaborate on that as well, because I'm not married.
Yeah, or any other type of relationship. I think open and honest communication is, I think, key, because you want to share your thoughts and ideas and then be very open and transparent about this. So I think this is very important, to actually see the different members of an international collaboration team as team members, as people working with you and not as potential competitors. Unfortunately, the science community, I have the feeling nowadays there's a lot of potential competitiveness because of scarce resources, funding, positions, etc. Right? But it is harmful for science and harmful for the projects if there's like this idea of potential working against each other. So doing this in an open and transparent way is super important. And I think this is the cornerstone of a good collaboration. Obviously, you want to collaborate with good scientists who know what they're doing, and it also helps to collaborate with people who are well organized. So if you collaborate with individuals who have a very good intent but are not well organized, this also usually leads to problems at some point, because there's deliverables to be and milestones to be kept to, and things you need to report to funding agencies and whatnot. And if your collaborators are not very communicative or disorganized and lagging behind, that always causes some sort of stress, which we want to avoid, obviously. So
yeah, it can. Okay, so I would say transparency, communication, and being well organized would
Yes.
would be a good start
Yes,
for
exactly.
collaboration.
Yeah.
So you have been a lab leader for over a decade now, right?
Uh,
What lessons have you learned about mentoring students who navigate these high-stress research environments?
I probably would say that yes, on the one hand, there is a high potential in everyone, independent on their initial starting point. Some students come in the lab and already know a lot of the techniques or the requirements that they need for running a project. Others will start a lot of things from scratch because they had never been exposed to this or that, and may see this initially as a disadvantage, but over the time I saw that this is not, because everyone can develop according to their own strength, and there's hardly anyone who is completely full-rounded and good at everything. So some people are really good at doing experiments, others really good at analysis, others are excelling in the writing and presentation of data, and you can foster these strengths in everyone, and basically also adjust the research project or the level of the research questions according to the strengths of a person. So that's why we're working in the team, so that not everyone has to do everything by themselves, but you can a little bit balance this, and then this way everyone can get really good results and manage to get their degrees or whatever they're working on successfully. The other thing which I would say one learns over the time is that the initial things that on a day-to-day basis are very worrying or stressful for an individual scientist, on a larger scale, one can say this is maybe take off the heat a little bit and say, okay, this is not that important. So the success of this one experiment, of this one readout, of this one measurement will not determine your career path or your future chances or whatnot. And to guide people in this and to help them see that what you do on a day-to-day basis can also have disappointments and setbacks, but it's the long-term goal that is important, and in the end we're working to gain additional knowledge, and in that case also maybe a negative result or insignificant result is a very important one, and to convey that to the students, that I think I've learned to try to apply over the last decade and more.
Sounds like the students in your lab are having a good time, and they're
I have to ask them.
Maybe we'll have a chance to ask them as well. But it really sounds like you are trying to maximize the potential and the people in your lab and in your team.
Yeah, I think this is what everyone should do, no matter in which situation you are, whether you're a scientist or in a company or having a small team, in all kinds of positions in life and all kinds of areas of work, seeing the potential of someone and trying to use or apply this potential well, and not dwelling too much on maybe the things that they're not excelling in, I think is always a good strategy.
What would be your advice for early career scientists?
Not to stress too much.
That's a very good point.
It's maybe easier said than done, but I have the feeling, I mean, this is again not maybe only in science, but the level of expectations from society, friends, family, and then what people put on themselves is very high nowadays, fueled by a lot of information you get via social media, the news, whatever. And this comes with a lot of potential anxiety as well. Or will I manage? And there's so much competition. So there's a lot of stressors in the life of young scientists, and some of them are real, some of them may just be perceived, but generally it doesn't help to worry about those long-term future stressors too much. And the important thing is to focus on the things you're doing right now, on the research question that you're addressing right now, and to doing this well. And if you're doing good research and finding additional, like, interesting knowledge that wasn't there before, this is what is most important at that moment. And everything else, I think, will follow suit. And it doesn't help to already plan 5 or 10 years in the future. What will I do in 10 years? And what do I have to do now to make this happen in 10 years? I think often this is overthought and too much. And what you should focus on was what you're doing now. And as a young researcher, it's mostly your research project that you're working on. And to do this well, I think, should be the main focus. And then it's also a bit less stressful.
So do your best and hope for the rest, I would say.
Yeah.
So thank you very much for joining us today, Mathias. It has been a great conversation. Thank you for your advice, for sharing your science with us and your career path. And we wish all the best to you. And we look forward to following the continuation of your research.
Thank you for watching part 1 of our special episode. Tune in for part 2.